Correct.
Good afternoon, everyone. I welcome you all for the Journal Club meeting of India chapter, Council for Ayurveda Research. And today we are blessed with the presence of a academician, researcher, and a clinician who is globally acclaimed, Dr. Asmita Wele. I will not waste any time more. I would like to start quickly because I need to give her more time. I'll just briefly introduce her. Many of you must be knowing about her work in research. She is a professor and head of the Department of Rasa Shastra and Vaishajya Kalpana from Ayurveda College, Bharati Vidyapit. She is also honorary professor from the University of Hungary, and her expertise lies in designing and execution of end-to-end research projects of Ayurveda formulations. Basically, apart from standard formulations of asvarishtas, telam, or herbal formulations, she has experience of making parpati, sindur, potli, and abrak sattva as well. She has been principal investigator for DST funded project and many Ayush projects, along with a member of Ayush International Cooperation Committee, who is involved in scientific research at Gujarat Ayurveda University, Jamnagar. She is also a BOS member, academic council member at BHU Varanasi, and delivered more than 125 lectures on various topics of mainstreaming Ayurveda across all the verticals. She is a guide for many MD and PhD students, published more than 40 research papers in various journals, and currently she has a very unique training achieved, and she is acclaimed reviewer from Scopus Elsewhere certification research agency. It is Elsewhere reviewer training program, and now she is a certified reviewer. Our journal club activity is basically focused on how to decode a research article, how to read more about the research in between the lines, and when you want to publish a research, how to ideally draft it in a scientific manner. So I think there is no other person who can clarify both the aspect in terms of a writer and in terms of a reviewer, and take this opportunity to interact with her. Today's presentation topic is physicochemical analysis of herbally prepared silver nanoparticles and its potential as a drug bioenhancer. This topic itself was suggested by Madam, keeping in view many experimental research possibilities for Dravya Guna Rasa Shastra people and students, and those who are interested to work in it but not aware how to publish it. So I would like to welcome Dr. Asmita Wele ma'am, and I really appreciate that you agreed to present today's topic and gave us your extreme time for the Journal Club meeting. Hand it over to ma'am. Thank you so much. Ma'am, your mic is off, I guess.
I think I requested you for just 2 lines. So
it's very difficult to put your profile in 2 lines, ma'am. Yeah.
I'm still a student, and I still get tensed when I have to talk on such topics where the experience of Ayurvedic literature is much more than what is going on in the current field, and many times I feel at loss of words when I come across such articles. So while after selection of this article and sharing it with you, the म्हणजे मराठीत म्हणतात तसं बाण भात्यातनं ऑलरेडी गेलेला होता. So later I thought, why I selected this article and am I really going to be able to present on it? Being a reviewer, yes, your brain works better than what you can explain. Many times you understand, you can assimilate, but you cannot express what exactly you have understood. And then secondly, this problem is faced by all the students across the pathies or across disciplines. It's not the problem of Ayurvedic students alone. It is the problem of all science students. They know, they understand, but how to express, how to write, especially in the writing, that's a big dilemma. That's a That's a field where everyone needs to put in efforts and everyone needs to refine it every day. So you have to write and read and rewrite and reread, and then finally you say, okay, now I understand this correctly, so probably others will understand. So that's why, as you rightly pointed out, this journal club activity is very, very important, where students can learn how to write and how what they have written will be analyzed by the reviewers. That's one thing. In the beginning, before I forget, I must thank Pratibha, Dr. Pratibha Shah, who is sitting in US and who has really created this awareness. I have seen no other person like Pratibha, who is um, who is a wonderful administrator, a good academician, and and a good clinician also, who knows what is needed for the field, and she's always in search of people who can help support her in that endeavor. So I'm fortunate to join her and you here, a representative in India, to participate in this activity. So let's begin. This is the activity for car, and let and you all are there to tell me whether I have succeeded or failed. I'm ready. I'm prepared to fail as well today. I don't know. Yeah. So this is the. Just a moment. Let me just do this screen. Huh. This is the physico. This article is physico-chemical analysis of herbally prepared silver nanoparticles and its potential as a drug bioenhancer. As this title tells you, we can pick up a few terms. This article is published in the Journal of Elsevier. Title of the journal is Open Nano. Uh, this journal is not a regular journal, which is studied or which is read by Ayurvedic fraternity, Ayurvedic students. This is something different than what they read. They generally read Ayu, they generally read Ancient Science of Life, they come across JAIM. But this one is different. And that's why I wanted to, I thought of selecting this article. Uh, this article has been studied by Shirisha Mukkavalli, Vijay, rather, this article has been written by Shirisha Mukkavalli, Vijay Chalivendra, and Balaram Singh. These three authors, they are all are from USA, the University of Massachusetts, Dartmouth. From there, from that institute, they have studied. And Dr. Balaram Singh also has a school of Indic studies. That's why this silver nanoparticle or Rajata Bhasma has come in picture. So when I first read the title of this article, I had this question that, okay, herbally prepared silver nanoparticle, is it Rajat Bhasma? Then the second, when I read the names of the researchers and the School of Indic Studies, then I realized that, okay, there is some connection with India and probably therefore some connection with Indian culture and connection with Ayurveda as part of that, and they must have studied on this Rajata Bhasma. The half part, drug bioenhancer, was a bit tricky for me because coming from the Ayurveda fraternity, I'm not used to think about silver or for that matter any other metal as a bioenhancer drug, because we know this Suvarna Bhasma, Rajata Bhasma, Tamara Bhasma, Loha Bhasma, these are our right and left molecules. So whenever a patient comes in, we might think of all these products coming out of it, and that's why this drug bioenhancer was a little bit. mm, uh, I was intrigued by this nomenclature, and that's one of the reasons. Now, there could be many research journals, many articles, but for a journal club, it is a custom that the article should be selected from a reputed journal having some index factor or source normalized impact factor, um, like SNP. It's written over here for this open nano. It is 1.6, so it's not a very high impact factor journal. It's kind of somewhere in the middle, and the site score for this journal is 6.5. So the journal is doing fairly well and that's why I went ahead. Though I like the article, I just checked the journal's authenticity or journal's ranking or status as well. So this is Elsevier Journal Index in Scopus with a SNP 1.6 and site score 6.5. This particular article, Physico-chemical analysis of herbally prepared silver nanoparticles, has been, it was published in 2017 January, available online, and since then, in last 3 years, it has received 8 citations and about 29 downloads, and that shows that this article has done a good work. So let's go ahead. This is silver nanoparticles. Why it is so? So the nanomedicines and their applications in modern diseases is the buzzword of current times. Cancer, Graves' disease, irritable bowel syndrome, many muscular diseases everywhere people are searching for cure. These diseases are yapya. People need to take care of their health. They need to keep taking medicines like in diabetes, like in hypertension, and there is no promised cure. So that's why it is that's why it's about search of new drugs continues, and therefore nanomedicine is the most important area that has come up. Then in nanomedicine also, the drugs. Why nanomedicine? When a drug goes to nano size, the dose is reduced and its potency is multifold, it increased multifold. So therefore, this article, therefore these researchers also were interested to study nanomedicine. Another important importance or another point which came to my mind about this article is there are many drugs from Ayurveda, including oils, which can be bhasmas are available in nano form. Everybody knows that, but there are oils also which are in nano form, and if that is the case, uh, people should work on it, and that's why maybe they have selected it. The gray area of Ayurvedic bhasmas, or for that matter, all the dosage forms, is that the ADME, absorption, distribution, metabolism, and excretion, is not known completely. And even there are various gray areas in dynamics. So people are studying, but still we do not know a complete picture of that drug as it is known today for any XYZ drug. So that's an area where there is a possibility of measurement, and that's where Rasashastra Dravyagun people can contribute substantially to understand Ayurveda. Not only for other people, but for us also. We need to know how our drug works. We know Rasavirya Vipak, we know this Rasapancha, we know Gunas, we know Karmas. But then we are always going arbitrary. We are always going
Excuse me, ma'am. We have lost your voice, I guess. Ma'am, you are not audible.
I'm not audible?
Now it is clear, ma'am.
Oh, okay. So with all of this, it's possible that we can definitely, or people are interested in this, and there is a space for us to work on these areas. Therefore, I have selected this particular article and not the clinical article for today's journal club. So there is a possibility of work. There is a many people are working. Many people have published also, but then we need to know. And as I said, as I briefly mentioned, or just in past, that the researchers are from biomedical engineering and biotechnology background, working in the USA. They got interested in silver nanoparticles. They were working on some bioenhancers. They got interested in silver nanoparticles, and that is why I thought, being an Ayurvedic physician sitting here, that what is so great that they want to know about this and how they want to use it. So that's why this article has been selected. So let's see how it goes. The objectives of this article are very clearly put. The researchers very well know that nanomedicines are of two types. There are drug molecules and there are carrier molecules. So, drug molecules, the main objective of that molecule is to deliver the effect. And the carrier molecule takes the drug to the site of action. However, it's necessary that the carrier molecule should not be toxic. First and foremost thing, it should be safe. And the second important thing is the carrier molecule must be bioenhancer. So it should reduce, it should enhance the availability of the drug after metabolism at the site of action. And therefore, the bioenhancers which are used today, which it's a huge field, vast field, and people have worked in it a lot. About 20-25 bioenhancers are known today, which are synthetic in nature. And therefore, modern scientists, the synthetic bioenhancers have advantages as well as disadvantages. So, specifically in case of cancers, we know that the drug is effective, but its carrier molecule is toxic, and that's why we cannot put it together. And therefore, a lot of research is going on. Money is put in, but it is wasted as well. And scientists are searching for carrier molecules from natural medicine, and that's where they look at Ayurveda. They are looking at Ayurveda here not for the sake of Ayurveda, not for Ayurvedic treatment, but they are looking at Ayurveda here for getting some potential bioenhancer molecules, and here the researchers are clear in their objective. So they are looking for a molecule which will enhance bioavailability of the drug at absorption. They want it that the molecule should not interfere with of the drug metabolism. It should have a similar action so that absorption will happen in less time. And in this, their assumption is that silver bhasma nanoparticles, being natural in being natural, they must be less toxic and they may carry out all these functions. Before going ahead with this, they put three clear cut objectives: the structure, composition, and nature of Rajat Bhasma, that is silver Bhasma, they have written. Then the second objective is okay, once you know the structure, composition, and nature, whether the presence of this Rajat Bhasma alters the physiological environment of the cell or not. That's their second objective. And if it alters, then what is the mechanism? Can it be found out? So, with these three clear objectives, this paper is written. In the methodology section, they have described very well about the product which they have selected. They have selected Rajat Bhasma, made by Baidyanath, a 117-year-old, maybe 20-year-old, 120-year-old pharmacy. They have taken the process of making from there, and then they have studied the Rajat Bhasma for its morphological, structural, elemental, and chemical nature. So they have characterized it using ADX technique, semidax, ICP-IS, TGA, and powder XRD. So these four techniques they have used to characterize the molecule. After that, they have conducted permeability study. So since this paper was shared with you, I assume that all of you have gone through the details and you have read the paper. So here they have selected intestinal epithelial cells which are grown on cover slips, and the cells are in monolayer. These two types of cell lines, CACO2 cells and HT29 cells, they have selected. They have also selected two markers, Alexa 488 and Alexa fluor 488, labeled deactivated recombinant botulinum neurotoxin. These two are the different marker compounds. They also used atomic force microscopy for mapping the changes on surface of the live cells. Right? So this is what is the methodology which has been explicitly explained. After these studies, this paper talks about the characteristics of Rajat Bhasma. The Bhasma is crystalline in nature. It is spherical. The particle is spherical. Its size is 350 nanometers. It's a complex, heterogeneous, non symmetric arrangement of atoms in the molecule. The, the final molecule is composed of silver, sulfur, carbon, oxygen, hydrogen, and nitrogen, in which silver is maximum, sulfur is lesser than that, carbon, oxygen, hydrogen are still lesser, and there are also arsenic, mercury, aluminum, and silica, which the researchers are saying that they are contaminants. The researchers have also identified the source of contamination, from where this contamination has occurred, especially in case of aluminum and silica, because while digesting the samples, while preparing the samples for these tests like semidax or powder XRD, we have to process the bhasma with some chemicals. We have to use different types of plates, and there is a possibility that some nanoparticles of other elements may have gone there. And another possibility, which they have very clearly explained, that the processing drugs during the process of making silver bhasma, rajata bhasma, Shodhan, Maran, the herbs which we use, they do contain mercury, they do contain aluminium, they do contain arsenic because of the chemical fertilizers which are there in the soil, which are there in the water, because um, we pour most of the times untreated water into our rivers, and then it is taken up for the fields. It comes in all the circulation, even we drink it. So therefore these are the sources, and they have tried to identify the sources. They have not mentioned the soil source, but they have mentioned the instrumental sources. There are certain additional findings which they have mentioned and which I will discuss in the last slide. For the first objective, as I said, that a clear cut characterization they have done, and they have also mentioned, they have compared this characterization with the standard data. For example, for XRD, there is a there is an international center for diffraction data, XRD diffraction data, and variety of XRD patterns are available in that center, which can be compared with the bhasma, and therefore we get a clear cut structural analysis, structural pattern of our bhasma. So they have compared this Rajata Bhasma against Ag2S, AgO, and Ag2O2 XRD patterns. They have matched it, and then they have analyzed and interpreted what is there in our Bhasma. They have conducted same to find out which are the animals and how the surface looks. Surface electron microscopy is same, coupled with EDAX. In that, they found out 4 molecules, mainly silver, sulfur, oxygen, silicon is there, and 2 more. But to reconfirm it, they have also conducted ICPAs, and this is a common and standard practice. When you want to know a molecule for sure, then you have to conduct a battery of tests. There are about 10 different tests. So they have performed only 4, not all. And that's one of the lacuna, but we'll come to that a little later. Uh, with whatever 4 tests they have conducted, they have interpreted it well. And therefore they are able to give us the structure of they write the structure of the molecule. After that, for the second objective, whether the bhasma can enter into the cell, if it enters, by which pathway it goes, whether it enters inside the cell membrane or it enters between the interstitial spaces of the cells. So that is paracellular pathway or transcellular pathway. These are the 2 major pathways a drug enters. They have studied it using two different cell lines. Now, these two cell lines, the CACO2 and HT29, both are interstitial adenocarcinoma cell lines, and they both are used to study these nanomolecules and the bioenhancers, variety of bioenhancers. These methods are standardized, they give you a correct understanding, and that's why these two cell lines are used. Of course, when we are conducting some experiments, it's necessary that we do the experiment with known models, known molecules, and then we can think and modify also accord to suit our need, and that's where innovation comes in. So here they have used the standard two cells. The cells were co-incubated with the bhasma, and then uh, this Alexa 488 dye was induced. Here in this picture which I have taken from the journal, you can see the first uh, film where there is only, these are the only cells. This is a standard control, so this is a mass of cells. Here in this picture B, you can uh, you can see some of the you see the dye in green color, where they have shown very clear cut epithelial cells, and after 1 hour, where you can see that the dye has started entering in the paracellular areas, and in this C you can see that there is a clear cut network. So the dye has reached at the end of 2 hours. So in the first group of cells, that is picture A, B, and C, control versus the cell line, CACO2 cell line after 1 hour of induction of the dye and after 2 hours of the induction of the dye, they have shown that yes, the Rajat Bhasma enters into the circulation on in this that single layer, and it creates some area. It passes through this paracellular mode. In the next study, what they have done is uh, 1 milligram per ml bhasma was treated for 15 minutes, and at 2 hours it shows well defined boundaries. So in this picture they have really clarified that this is a group of cells, mass of cells, which is unexposed. These are HT29 cells. So these are unexposed cells. You can see the mass. And here, the same cells after 1 hour, there are the regions which are partially exposed. These are the lines, and a different, well differentiated molecule, fully exposed regions to the bhasma. They have very clearly shown the boundaries, and at some places like here you can see the fibrous structures also. So the uh, topography, AFM, atomic force microscopy, the topography done by using this microscopy clearly indicates that transport is paracellular and it is in uh, time dependent mechanism from 0 hour to 24 hour, and at 2 hours interval this is maximum. That's how they have clearly mentioned in the results, and they have discussed the results. After reading this particular article, what I felt, there are many graphs, there are, you all, I assume that you all have read this article. So they have given a very nice graphical representation of XRD patterns, semi-DX tables, the same images. They have also given this comparison of control group and study group at 1 hour interval, 2 hour interval for both the cell lines, and the confocal imagining of the detailing of all the methodology is also mentioned. So this is a good paper in a way. I find whatever they have focused, they have delivered. But from the Ayurvedic physician's viewpoint, and that too coming from Rasa Shastra, When I know that Rajat Bhasma is prepared in so many ways, there are so many, it's not a single way, it's Parat Gandhak Marit Rajata Bhasma. Then there are some other elements. There are about 14, 15 variations of making Rajata Bhasma, depending on the type of putta, depending on the ingredients which are used to triturate Mardan and Bhavana. For that process, many things are there. Even for the first Shodhan, we use 5 different ingredients, Taile, Takre, Gava, Mutre, Haranale, Kulattaje. And then uh, those are responsible for the development of our molecule, that is Rajata Bhasma molecule. Now, this information and the chart which they have shown, I'm sorry, I should have put that chart here. The flow chart which they have made, it has some major shortcomings, major flaws, because they have written in the chart that the bhasma is filtered somewhere, huh? After heating, after gajaputa, it is filtered. So it is possible that in pharmacies, when the bhasma is not properly prepared and there are big molecules, it is filtered through sieve, and the filtrate is taken for further processing, and the top molecules are left aside. So that's a process, but that is not a standard process. In a scientific process, it is not expected that these molecules should be there. That's one thing. They have also not mentioned the bhasma tests. One line would have been sufficient. I don't expect that they should write a whole full battery of bhasma test, but this bhasma had complied Ayurvedic bhasma test that the researcher should have mentioned. Because the bhasma comes from Baidyanath, it is very possible, it was easily possible for them, and the company is really good company, reputed company, so there is no chance of any compromise of on the quality of bhasma. They should have written that. Uh, Probably the company should have, they might not have provided data, I don't know. There are gray areas here, but that information is not there in the paper, and that's what documents speak for. So when, as a reviewer, when I look at any paper and I do the review of a paper, the person is not there to explain. Document speaks for itself, and when document speaks for itself, these points, they should be taken care of. That if you have not written, if I have not mentioned, and that's what reviewers check. So characterization with more methods, I said that there is a battery of about 9 or 10 different tests, which gives a comprehensive structure. It would have been added more value to this analysis. I also understood, I learned new things here after reading this paper, that this Rajata Bhasma can be a bioenhancer for various drugs. Now, these people have tested its activity as a bioenhancer. They have mentioned that this Bhasma food, this is a new area of thinking for me. That when I'm using Rajata Bhasma processed with Agastipatra Swaras, Rajata Bhasma processed with some other Swaras, Triphala, Kvat, will it be efficacious in the same disease, or will it be efficacious in the other disease? And whether I should look at the Rajat Bhasma as bioenhancer, or I should look at the Rajat Bhasma as the main drug which is responsible for delivering me the effect. So this was a new thought process this paper has started for me, and we have to see now how to look at our Rajat Bhasma. We can look at from both the angles. Where it is in very small amounts, we might say that this is acting as bioenhancer. When that is the main drug, then it might be a different calling. Uh, 2 points which I could figure out, which the researchers also have mentioned as new leads, that how this bhasma works in the cell membrane, with the cell membrane, whether it binds to the receptors, whether it enters inside, what exactly it does. And within cell, there are a cell is a a group of many small structures. There is reticular endothelial system, there is mitochondria, there are many structures. So how this bhasma reacts there, there are various models, and we could use them for one or two of our bhasmas to do another studies. So mechanistic studies can be done using these models. I'm coming to the end of this analysis of this paper, and I from after studying this paper, these are my impressions. This I came to know about a new or different aspect about Rajat Bhasma. Characterization has been clear, whereby they have come out with a specific understanding of the molecule, that the Rajat Bhasma molecule is spherical in shape, its size is 350 nanometers. It is a molecule of multiple atoms in a heterogeneous, which are distributed in a heterogeneous manner, and there are various phases of this crystalline molecule. So the characterization understanding is good. They have studied for the mechanism with two cell lines, CACO2 and HT29, for mechanistic checking. What happens is, many a times, due to variety of reasons, and mostly due to lack of resources. By resources, I do not mean money only. I mean availability of doing this kind of experimentation. So lack of resources, we even our PhD students just do one or two studies somehow, and they get away with it. So to confirm any of your hypothesis, you need to verify the mechanism not only with one, but at minimum two and more, maximum five different cell lines. So here it is cell line. If you want to confirm a toxicity of your drug, it is expected that the toxicity should be conducted on at least two types of rodent species and a type one or two types of higher mammals, and then you confirm the toxicity. So similarly here they have done this mechanistic checking with two cell lines. They have also used two different markers, alexa fluorophorate and dr botulin a, for pathway confirmation. So they have not stopped at one mechanistic approach, but they have also cross-checked it. But they have also cross-checked with against time. So at 1 hour, 2 hours, 24 hours. So what is the exact concentration? What is the time to reach exact concentration of Rajat Bhasma into the paracellular spaces or extracellular spaces? So this way, the study is foolproof. One more important thing is the supplementary material for the standard methods is provided with the article. So those who are interested can click on the supplementary material link, and they can get how the experiments are conducted in detail, how the cell line is mounted on the cover slip, how it is grown, what type of materials are purchased, how the MTT assay has been done. So micro details are there so that this experiment is replicable. Anybody can repeat it and then compare the results of this repeat experiment with the experiments, which the results which are presented in this paper. So in a way, it's a well-designed study. There is no confusion and loose ends. Whatever objectives the paper has set, the paper has met the objectives, main findings. There is a focus on the main findings, and the researchers are well observant researchers. They have put the additional finding, which is not intended, but it is observed that this Rajat Bhasma is non-toxic in contrast to pure silver nanoparticles for these cells. This finding was important for them as well, because they are looking at it as a bioenhancer. They are not looking at it as a potential drug molecule. And for a reader like me to know that my Rajat Bhasma is non-toxic as against pure silver nanoparticles, it gives me more confidence to use my drug in my patients. So this is what is the contribution of a research article in the world of science. So I think with this I'll stop here, and you go ahead with questions and happy to answer any questions, and let us take ahead this discussion, because as I told in the beginning, even for me, studying this type of articles is a new learning, and I like to learn. So let's discuss. There could be many more points coming from you. Thank you very much, Pranita. Thanks.
Thank you so much, ma'am. And I must appreciate the whole topic and the way you presented it. Sometimes we think that experimental research is alien to Ayurveda. And as you rightly pointed out, bioengineers, biotech people are doing Ayurvedic studies. Now it's high time to really look into this area where we can contribute a lot. And I believe that like the main objective of resources which you have mentioned is also very correct, because man, machine, and money is the basic resource, but mindset is the ultimate one. I believe in that mindset needs to be developed. And today with your presentation, I hope that many of the participants, they are representing various colleges, they are coming from various background of UG, PG, PhD researches, and there are someone are practitioners as well. So they will take that mindset out of this presentation, that okay, this can be also done. And with the unique way of presentation, you all have been experiencing that IMRD format Madam has focused. Introduction, material methods, results, and discussion. And there is one more aspect to it, impressions from the article, because every reader takes out something new from the same article. And once you read like good papers, a good reader becomes a better writer. And that's whole the purpose of the journal club, because with journal club we discuss paper, we read out the coding of the paper, which the mindset of the researchers, and then we understand the possibilities. So maybe a good research paper can also develop a good hypothesis. And I would also like to mention a disclaimer that journal club activities are purely academic activities. We are not intended to judge or criticize any researcher, but we want to showcase their work in a different light, in a light of scientific review process. So I believe everybody must have gained a lot of insights on this topic, and we are open for questions. You can put it in the chat box. I will read it out for you. There is one question, I guess, from Amit, Dr. Amit, that explaining tedious article in simple way is appreciating. And can you please tell us about the two markers that authors used, Alexa Floor 488 and Dr. Bond
Uh, hello, Amit. Good to have you here. I also see a comment from Parth Kale. It's privately written, but first I'll answer Amit's question, and then I'll go back to Parth. Okay, Amit, in this area of markers, I am as ill-informed as you are. I know that this Alexa Floor 488 is a dye which is used, it is a radioactive dye which is used to trace the movement of a molecule. So you have to, so this particular dye is attached to the bhasma. Now all these are chemical terminologies. So what attachment means, when we triturate our rajat bhasma with some ardrak swaras, we are attaching some of the molecules, phytochemicals of ardrak to that silver atom. It's the same way. Here only thing is this Alexa floor is fluorescent. 488 is the name, is the label of that, is the name of that molecule, and that is tagged to the silver atom. Therefore we can see that green, the fluorescence, green fluorescence is seen on the X-ray plate, and that's where we can understand where our bhasma has gone. Now this botulinum neurotoxin is a another molecule. It is actually a drug molecule, but it in small amount it works as marker, and it goes to particular, it also goes in a particular cell. So it also gets attached to this Rajata Bhasma. So these are the two marker compounds which are attached to our Bhasma molecule, and then we can trace them using this XRD technique, or the photo. Just a moment, give me a minute. I told you, even I am not very well conversant with these things. This is the uh, AFM topography. So they have done this AFM topography. Atomic force microscope is the instrument which they have used. Yes, I studied. I have, I have searched some of these microscopes. You can search them um, on Google, and you will be able to find the pictures of this machine and instrument. And in that instrument, you take the photographs. Also, it's called confocal imaging. So these are the techniques which are used in chemistry. So chemistry and biotechnology, these two fields are together, where they have where they have studied this particular drug, and these two, using these two marker compounds. Coming back to Partha says that Ayurved Prakash quotes Raupya Bhasma should not be used as single medicines except Shvitra. So can we think that Raupya is carrier molecule in most of the indications, and it is possible that as carrier it does not become toxic? As individual drug, it may show some adverse effects. Perfectly right, Parth. Even I am thinking the same. You have given words to what I was thinking. That yes, we do not use Raupya Bhasma singularly. Siddhinandan Mishraji has mentioned that you should not use Raupya Bhasma singularly except Shvitra. There are a few papers which on Bhasma, which have quoted this particular reference which you are talking about. So I would like to share those with you separately some other time. But yes, what you say is perfectly right. And these type of questions you should ask, so that we can take up some study. Rajat Bhasma for Shvitra can we find in experimental also, and in clinical setting also. Only thing is we have to be very precise about our methodology, just like this research paper has done. That's what I feel. That Partha has also put this question on the platform, but privately. So
Okay.
yeah.
When
And,
to the extension for that same question, there are so many methodologies in Ayurveda, particularly how the medicine works. And if you are working on characterization, the whole domain changes to the biophysics level. So, how to write methodology in such scenario as a writer, a first-time writer, you can say, who has already completed the research, but now you have also pointed out one area where we can express our methodology in terms of flowcharts, diagrams, tables, so it becomes more readable and understanding for the public, those who are not directly working in like as there are so many markers and other things which we are also are not aware. So how to write it in the methodology, can you just brief on that?
um, yes. See, when you are writing a methodology for any experimental study,
Yeah.
you have to give all the details about the process, what you have done. And as this paper has done, there should be a flow chart.
Yeah.
In our papers also, we generally like to give flow charts and uh, pictures with legends and the
Hmm.
correct names below.
Right.
What happens is, many a times I see papers where only text is written, there are no charts, there are no figures, no schematic diagrams. And if there are figures or flowcharts, uh, if there are figures mainly or photographs, legends are missing, or the titles, labels to the figures, those are also
Hmm.
missing.
Hmm.
And then that conveys the reader, very poor information, that
Yeah.
gives reader a
Yeah.
poor, and that should be corrected. Now, if you are writing methodology for clinical study, there are various guidelines, even for clinical study of observational types or randomized clinical trial. there are guidelines like Prisma, there is a guideline of Consort. In those guidelines, flowcharts are given. So we have to use that flowchart for writing the methodology so that we don't miss out on anything. You have not asked, but Pranita, let me clarify one thing on my own. Um, After reading many research papers, I have realized that uh, there is a confusion between research about drugs. The clinical person starts with the drug standardization and he gets confused.
Yeah.
Ayurvedians should stop it. We should stop it. We should take the drug as drug, whatever from wherever source it is coming, and the company, the details of the drug, there is a standard drug dossier file. So if you are doing a clinical research, I think your focus should be on the clinical outcomes, the methodology. You have to follow rigor, you have to maintain all the charts. Follow-up should be on time. You have to maintain the diaries whether the patient has taken the medication or not. If you are doing some biochemical parameters, then interpretation of those parameters, putting the whole data together, discussion on the results should be a priority. Don't enter into the details of the drugs. That's where students get confused and misguided.
Right.
They spend more time on the drug standardization, and then they lose the interest. As it is for clinical trials, that is not their main focus. Uh, Any person who is sitting in the clinic will think of, I want to use Rajat Bhasma or Gandhakrasa and I want to use it. That's it. If it gives me result or not is my concern. Similarly, a person sitting on the joint between clinical area and uh, reservation like me, we are always concerned about the quality of the drug, what exactly the molecule is, how it is going to work, new development, new drug designs, and that's where we should focus on the experimentation. We should focus even on clinical experimentation,
Yeah.
because when we are working with different suppose there are 5 different rajata bhasmas come in front of me or we make in our department, their efficacy will be different. So we need to work with the clinicians and we should tell them that okay, this rajat bhasma is prepared using the standard method. For this rajat bhasma I have used jyotishmati, so
Hmm.
can you use it in psoriasis and give me the results? Maybe the patients only 12 or exploratory study, you can do it in very small number of patients, but the questions should be precise. Like in this paper, they have precise focus questions, they have answered those questions, they have correctly written the methodology because it is experimental. Coming back to you, how to write the methodology for any experiment, because you are not aware of these markers and dyes and this and that. Enough information is available in good journals. We do not, or we should not rely on hearsay. If we meet a person, somebody comes to me or to you, or I go to my colleagues here in biochemistry, in pharmacology department, uh, even in various institutes, various other institutes who are working with chemistry, I go there, I talk to them, and I take the impressions and I start working. That's where we go wrong. We should talk to them, we should get the information, but then come back home, type it,
Hmm.
check the real method, what the method has come, research, check research papers, or read 2 or 3, at least one good review paper which has given that method, which has written that method, and then write that method for our for my own outline of research work or methodology. That's where the thing is. We don't do this exercise, so many reasons, and then when we complete the study, in the presentations we say that that madam has told me this method and so I did
Yes.
it. That's not a way
True.
of science. Ayurveda, Charak Samhita, Rasashastra, no one teaches us this. They all teach us to go to the roots. Mule kutha raha. So go to the root of the method, figure out, find out. Again, discuss with that person if you find some problem with that method. then go work with the person who is doing this for you, and you will be able to write correctly and exactly.
Yeah, true. Very important aspect you have highlighted, that a lab research should contribute to the clinical insights. And if our decisions are based on data and data which is coming from the experimental research, our decisions are more accurate. And that is when we can call it as evidence-based clinical research. Maybe that will be the golden era where we will have exact methodology for all the basic fundamental drugs. There is one more question from Dr. Ajit. We'll take it, ma'am.
Huh?
Can you please explain about atomic absorption spectroscopy, which is mentioned for plasma?
Yes, Ajit. This atomic absorption spectroscopy is a refined technique to understand the elemental composition. So, when we make bhasma, we are actually making the pure element impure, especially for dhatu bhasmas. We are making the pure copper, pure silver, pure gold, which is 24 karat gold. We are making it impure chemically, but we are making it very effective and non toxic for use as medicine in our body. So that's where we need to understand the structural change and compositional change. That's where we do atomic absorption spectrophotometry or spectroscopy. There are many techniques in spectroscopy. There are many techniques in chromatography. Which technique is to be used where is very precise, and there are many examples and studies which have been conducted. So, if you want some more information, you can refer some good articles, or now here, Pranita, I can say that they can refer a chapter in my book for a general understanding. I have written this book on principles of research methodology for Ayurveda. So in that, I have given this drug research chapter, different ways of doing it, and just mention these techniques. But there are many, many, many good research articles are available on atomic absorption spectroscopy, also other techniques. FTIR, for example, is a wonderful technique which should be coupled with this ICP-AS or AAS, because that gives us the role of our Mardan Bhavana with Kumari, Bhavana with Haridra, Nirgundi Swaras Haridra Yukta, or Kanchana Swaras Bhavana. What all happens to that molecule of our Bhasma is clearly understood by FTIR, because the organic groups which are coming in, they are seen. And similarly, there is a huge database of organic molecules. So just like this, Shisha has used IDCC, we have the FTIR databases. So you can just match fingerprint and I mean overlap and match which are the organic compounds which are there in your vessel, and thereby it's very easy to understand. Um, I don't know how much time permits here, but
Yes,
yes, I
yeah.
will definitely go ahead with one thing. I will definitely go ahead. हे जरा गरम करू मैं. With one thing that uh, different drugs when we use for treatment of or for making different basmas, the size of the molecule and shape of the molecule changes. So Suvarna Bhasma can be spherical, Suvarna Bhasma can be triangular, Suvarna Bhasma can be octagonal, it can be hexagonal, and it can have different organic groups. Sometimes it can have polyphenols, it can have mm, the other groups, flavonoids, it can have oils. So it's a complex molecule that we are making.
Yeah.
And Vaidyas who are practicing, Vaidyas who are making their own medicines, should stick to one method and should not compromise on the expectation or on the text which is written there. If it is written that this has to be dipped for 7 times, one should do that, because we do not know what the end product is, and therefore we should if it is written that this particular moola is used, then do not please use substitute. because that moola has a specific combination of various phytochemicals, and that is expected to sit on that to get attached to that silver or gold or iron, and therefore it's going to go and work there. So if we want to but now I think it's the mission of me that uh, yes, all what is written in Rasa Shastra text, whatever formulae are designed, developed, those should be followed uh, religiously. I'm using the term religiously, those should be followed. The manufacturing method with no compromise. Do not compromise on the Bhasma test. But on the other hand, start analyzing everything using the new techniques. And when you do this, when you publish these articles, you give confidence to newer students of Ayurveda. So today is the situation that only 4 out of 100 are practicing Shuddha Ayurveda using Rasavasmas or whatever, or all our formulations. What about the 96? So if we want to give confidence to all of them and good results, then there should be no compromise on making of our medicines. And on the other hand, our students should start working hand in hand with biochemists, with molecular scientists, with molecular biologists to understand what is going on with the bhasmas, how we are making it better drugs. And there is a lot of scope for patenting. If you make a bhasma which is not in practice today, which is, and you can modify it a little bit and you can show its action on a disease like psoriasis, which is not exactly written in Ayurveda. There is some simile, but not the exact picture. Whereas psoriasis has an exact picture, and if you show this, you can patent the drug. You can you can you can be a entrepreneur. You can be a good researcher. You can earn money out of it. So that should be done.
Right.
But for that we need data.
Yes.
And for that we need good research, and for that we need good publication. So you do good research, start with good questions, adopt a good methodology, publish without fail. Do not. I will tell you, it is really painful. There is a word, no, burning your bottom. That is true. Burning the midnight oil
Right.
is very, very important when you write, when you read, when you work with this. So even today, I can show you these papers. I was working. I have literally studied line
Yes.
by line on the methodology. Many things
Yes.
were new to me. Even now, many things I don't understand thoroughly, fully, confidently. But yes, this is the way. Nobody learns anything without putting in efforts. Yeah.
True. Thank you so very much, ma'am. In fact, you have concluded all my views about today's lecture, and it is
I
wonderful
will.
to listen to your work. And as you rightly pointed out, we are a student of Ayurveda ultimately. No matter how many years we put into it, we are still learning so many things. And your lecture is always brainstorming for others, and we learn every time something new from you. It's wonderful to have you. And I know 1 hour is very short to be in touch of your extremely wide scope of work. But I believe that today's attendees have learned so many new things, and they can write back to us on Ayurveda Research USA at Gmail. So whenever they have any question about this particular meeting or in future also, they can anytime contact us, and they can contact Dr. Wele ma'am also. Today's presentation is one of Feather in the Cap, because journal club meetings are always focusing on the type of research articles and how to decode it, so that you can write better articles yourself. And as Madam rightly pointed out, a good reader, good writer, good researcher, and ultimately a good Ayurveda clinician can bring up something new to the society, and not only to the society but to the science also. So I believe you all enjoyed today's meeting and write back to us. Looking forward to more presentations from your side also. And I would like to appreciate Dr. Wele ma'am for accepting our request to be here. And she is always there for Council for Ayurveda Research. And we are very pleased and honored to host you, ma'am, today. And I would also like to thank Dr. Rani Gupta, Dr. Abhishek Lulla, my teammates for Council for Ayurveda Research General Club Ayurveda Chapter in India. So thanks a lot, everyone. Because of you all, this meeting was wonderful and it was a fruitful discussion altogether. Thank you so much for being there. Thank you. Namaste all.
Thank you. Thank you everyone for patient hearing and keep writing to me.
Thank you, ma'am. Thank you so much.